Hi everyone. I'm Yanyu from Stanley Chi Lab at Stanford University. Today I'm glad to share with you our story of developing a live cell DNA imaging method called Oligo-LiveFISH. So the question we are interested in is to study the enhancer-promoter interaction and its relationship with transcription in live cells. And to achieve this goal, so we need an imaging technique that can image any region, including repetitive and non-repetitive region with a high signal-to-noise ratio. In addition, we hope our technique will have a high spatial and temporal resolution. And importantly, we hope this technique can be used in primary cell. However, currently, no method meets all these needs. So several years ago, our lab has developed a technique called CRISPR-LiveFISH, and it is a reagent-based live cell DNA imaging platform. So it combines two technology, which is FISH and CRISPR live cell imaging. So many of you are very familiar with FISH, fluorescence in situ hybridization. This is a golden standard for the DNA imaging. However, FISH need to fix the cell, so there is no dynamics. It cannot provide dynamic information, and also because the fish has very harsh conditions, so it probably will disrupt local chromatin structure. And also there is like CRISPR-based live cell imaging. Basically, we can construct a cell line and can express the dCas9 labeled with GFP and also the sgRNA. So they will specifically bind to the specific chromatin site and can be visualized under the microscope. And CRISPR-LiveFISH is basically combine the advantage of these two techniques. And what is CRISPR-LiveFISH? So basically, we will have three components, dye-labeled CRISPR RNA and a tracer RNA, and also a dCas9 protein. So all these three components can be purchased and prepared in vitro. And if we first anneal this CRISPR RNA and the tracer RNA, and ensemble it with this dCas9 protein, we will form what we call this FRNP, fluorescent ribonucleoprotein. And we can deliver this FRNP into the cell by, for example, electroporation. And once this FRNP is delivered into the cell, we can visualize it under the microscope. There are several advantages of this method compared to traditional methods. So first, there is no genome engineering, and so it can be allowed to detect primary cell DNA dynamics. For example, here you can say that we use it to detect the chromosome copy number in normal cells, which they show 2 chromosome copy, and also those derived from patients, which have 3 chromosome 13 copies. And it also have higher signal-to-noise ratio compared to using the dCas9 eGFP method. And there are two. We can prepare two types of the guide RNA. The first, you can using the CRISPR RNA and anneal with a tracer RNA. Alternatively, you can directly using a sgRNA, the single guide RNA. However, the first generation of LiveFISH is limited to repetitive region, which largely limits its capacity because most of the interesting genomic region are non-repetitive. So here, our goal is to expand the LiveFISH to image any region, including repetitive and non-repetitive region, by using a pool of a dye-labeled FRNP. So how does it work? So first, we will have a computational algorithm to design the guide RNA and use it to tile the region that we are near the region of interest. And then we will synthesize a pool of DNA oligos. And then we will do in vitro transcription to turn this from DNA into RNA. And then we will using some our chemistry method to label this RNA oligo pool with dye to form this FRNP pool. And then we will deliver it into the cell by like electroporation, and then we can perform imaging. So after that, we will track, analyze, and do mathematical modeling. So this is a basic pipeline. So once we have a live cell imaging method, the first thing is to confirm its specificity. So we're using the fish to confirm the specificity. So because the harsh condition of fish might disrupt the live fish signal, so we're using this sequential live fish method. So basically, we put the cell on a gridded cover slip so that we can find the same cell after fish. We first image using the live fish, and you see here, this is a signal. And then we perform the harsh fish condition. And after fish, you will see some of the live fish signal is already gone, but since we already recorded this signal in advance, so we can still compare that with the fish image, and you can see that they will have a very good co-localization. And then we expand this to multicolor. We're using 3-color fish to confirm the 3-color live fish, and you can see they have to target 3 different chromosomes, and you can see there is very high specificity. So next, we investigated the designing rule to design guide RNA for robust imaging, and we built an algorithm to help this design guides. So we choose a non-repetitive region, which is nearby the non-repetitive region, and we design different number of guides to tile this region. So as you can see, we designed many different number of guides like 12, 24, 48, 96, and 192, different number, different position of the probes to tile the same region. And we wanted to know what is the optimal number to image a non-repetitive region, and what are other properties that are important to get robust chromatin imaging in live cells. And we found that first, as few as 24 guide RNA can very robustly image a non-repetitive region. And here, as you can see that this is a non-repetitive region using 24 guide RNA, 24 probes. And also we wanted to characterize what chromatin features determines SNR. So we're using different probes and characterize its signal to noise ratio and the detection ratio. And very interestingly, we found that the SNR correlates positively with the chromatin accessibility, which is the ataxic or the H3K27 acylation intensity. And we further developed a machine learning model to characterize this feature. And the machine learning model also indicated that the most important property is probe for get a good imaging for good imaging quality is a probe number and the chromatin accessibility. So this is not only useful for chromatin imaging, but also maybe for the chromatin for the CRISPR-based perturbation. So later we developed this website to help to facilitate the other user to design their guides. So as I said, the most important advantage of our technique is that it can be applied in primary cells. So we apply this to a primary T cell, and you can see the dynamics of the activated primary human T cell. And very interestingly, sometimes we can see this doublets, which might be the sister chromatids after the DNA replication. And I also want to mention that it also works in other cell types like iPSC or iPSC-derived neurons. All these are very other cell types, which is very challenging to image using the previous imaging method. So next, we expand this to super-localization Oligo-LiveFISH to increase the localization precision to 10 to 20 nanometer in collaboration with Professor W. Moller lab at Stanford. So many of you might be familiar with this diffraction limit in conventional microscope, which is roughly lambda over 2NA. The lambda is wavelength, and this is the numeric aperture of the objective, and that is roughly 250 nanometer. And however, so what does this mean? It means that for the two objects whose distance is closer than 250 nanometer, their point spread function will overlap, and we cannot resolve them. However, if the molecule, the emitter, are isolated, we can always fit their point spread function with a suitable function like a Gaussian function, and their localization precision can be much smaller than this diffraction limit, and it will be roughly this diffraction limit divided by the square root of number of photons collected, and this number will be much smaller than 250 nanometer. And here you can see that using the conventional microscope, here is how this loci looks like, and the scale bar is 500 nanometer. But if we using this super-localization method, it will be the scale bar is like 50 nanometer. It's much smaller. So then we're using this super-localization oligo-LiveFISH to track a non-repetitive force promoter region with very high spatial and temporal resolution, and with 18 milliseconds per frame, and with like 10 to 20 nanometer localization precision. However, as you know, like actually the DNA inherently moves in 3D instead of 2D. So, for example, our collaborator Monolab has previously shown that for a trajectory diffusing in 3D, if we're using 2D method to characterize their dynamics, so because the 2D is a projection of the 3D trajectory, there will be roughly 30% error in estimating their diffusion coefficient. So to capture the dynamics in 3D, we use this 3D double helix point spread function microscope developed in Monolab. So how does this work? So basically, we will usually the loci in conventional microscope is one lobe, but here it will be splitted into two lobes. And the center of these two lobes tells you the XY position. And the rotation angle of these two lobes tells you the Z position. So without Z scanning, we can directly extract XYZ position simultaneously, and it is fast and reduce the photo bleaching compared to the conventional Z scanning method. So we're using this 2D and 3D super-localization oligo-LiveFISH to capture two color imaging of two sites simultaneously in live cells. So their genomic distance is 28 kb away from each other, and here is their typical trajectory. And we calculate their mean square displacement, the MSD, and the MSD curve indicates they are undergoing subdiffusion. However, you will see that their actual 3D distance is roughly 100 nanometer. This is higher than their 2D distance, which makes sense, right? Because the 2D distance is actually a projection of the true 3D distance. So then we will using this oligo-LiveFISH to get some applications. So the first one is that we characterize we're using the velocity autocorrelation to characterize the dynamics of chromatin movement. So here, what is a velocity autocorrelation? So basically, we will have so basically we will have calculated the velocity at time t, and then we will calculate at later time t plus tau, and we calculate how these two velocity correlate for one loci. And if they are negatively correlated, so they will have this negative peak, and that is exactly what we observed. We found a negative peak for their correlation function. So what does this mean? This means that for a chromatin, whenever it moves forward, they will have a tendency to be pushed back in their next frame, in the next time frame time. This is due to the elastic property of the chromatin, as well as a viscous property of the nucleus. And our results is very consistent with the fractional Brownian motion model reviewed previously. So we also use this to study DNA communication. So what does this mean? So we wanted to know when one loci moves, how fast another loci will feel this movement. So we're using multicolor oligo-LiveFISH to study 4 sites. So 1 and 2, 2 and 3, 3 and 4, like 1 and 2 are 28 KB away from each other, 2 and 3 are 200 KB away from each other, and 3 and 4 are 2 megabits from each other. Like intuitively, we will expect that 2 and 3 will be closer than 3 and 4 in space. And that is exactly what we observed for part of the loci, like roughly 2/3 of the loci are consistent with this expectation. And we call this canonical group. And you can see here 2 and 3 are closer in space compared to 3 and 4. Surprisingly, we also found 1/3 of the group is opposite. So the 3 and 4 are closer to each other in space, and the 2 and 3 are farther away from each other. So when we look at the Hi-C map, we do find that there is an architectural stripe between 3 and 4 that brings 3 and 4 together. However, the Hi-C only provides a static picture, but our method can also provide the dynamics. So we later, using our method to quantitatively analyze the organization and the dynamics of these two groups separately. So for the canonical group, we found that their pairwise, we characterize their pairwise distance, and we found that the square of their pairwise distance for the canonical group is very consistent with the Rouse model. Basically, the square distance scale linearly with their genomic distance. However, this is very consistent with the Rouse model. However, this was totally breached for the variant group. And as I said, our method not only provides the distance as revealed by the static image. Because we have dynamic imaging, we can also add a new dimension, which is velocity, to characterize how loci correlate with each other. So we wanted to know when this loci move, how fast and other loci can feel the movement of this loci, and we call this chromatin communication. So what we are using is velocity cross correlation. So basically, this is a correlation like before, but it is not the autocorrelation. It's a correlation between two different sites. And then we using a theoretical Rouse polymer model in a viscoelastic environment, and using this theoretical VCC curve to compare our experimental VCC curve and extract this relaxation time, Rouse relaxation time, or you can think of it as a DNA communication time between two sites. And we found that for these two sites, 28 KB away from each other, their communication time is 0.4 seconds. So basically, when loci 1 moves, after 0.4 seconds, the loci 2 will feel the movement of loci 1. So we then using this to characterize the communication time for other site pairs. And it is very interesting. We found that for the pairs which are the short distance in the canonical group, this our measured communication time is very close to what predicted by the Rouse polymer model. However, for larger distance, for example, 3 and 4, so the predicted their communication time will be 2 hours, but our measured communication time is much shorter than this value. It's only 380 seconds. So we speculate, instead of transduce their communication through the protein polymer, there might be another type of chromatin communication at long genomic distance that facilitates the chromatin communication. So basically, we think there are two types of DNA communication. One is through this DNA polymer, but there will also be some bridging complex, which could be protein or RNA, that can facilitate the communication for sites which are at longer distance, like the 3 and 4. They can directly communicate through this bridging complex. So the last example is to study the force enhancer promoter interaction and its relationship with transcription. So FORCE is a immediate early gene, and it transcribes very fast after stimulation. And we wanted to know why. So the static assay usually can map the enhancer promoter contact, but it cannot reveal the dynamics and also the cell-to-cell heterogeneity. So our multicolor live cell 3D imaging method can allow us to watch how the force enhancer and the promoter correlate with each other in real time. So basically, we study the force gene enhancer and the promoter dynamic when there is no, in normal growth, there is no stimulation, and also under the stimulation by the zinc, by the heavy metal. And we wanted to study how their dynamics is different under when the force gene is transcribed and is not transcribed. So here is how it looks like. So here is a representative trajectory of force promoter and force enhancer in 3D. And as you can see that when the force gene is transcribed, is on, so their trajectory is more compact compared to without zinc. And the variation of the trajectory in the XYZ dimension are also much smaller compared to without the gene transcription. And just pay attention that the scale on the y-axis is different. Here is much smaller. And also the enhancer promoter distance when the zinc is on is also shorter when there is no, when the zinc, without zinc. So then we using this to image many cells, and we found that, so first, when the force gene is activated by zinc, the enhancer promoter distance is shorter. So this is not surprising and can also be revealed by the static assay. However, we found that not only the distance is shorter, but also the distance variation is much shorter. As you can find here, the distance variation, how the distance changes, is also much shorter when the zinc is added. And also, we using the MSD and also MSCD, which characterize the dynamics for these two, for the enhancer and the promoter. And we found that their dynamics after zinc is much, much slower compared to without zinc. So all these results indicate that there is a slower dynamics for the force enhancer and the promoter during active transcription. So basically, we proposed that when there is when the gene transcription is on, for the force, at least for force gene, their enhancer and the promoter will have slower dynamics and will feel more confinement, will be in a more confined environment upon activation. So in summary, we developed a reagent-based live cell imaging method called Oligo-LiveFISH that can image non-repetitive genomic regions in live cells. It can allow primary cell detection, and it have high SNR and have high spatial and temporal resolution. And we use it to study the DNA communication, DNA dynamics, enhancer promoter interaction, and its relationship with transcription. So finally, I would like to thank Professor Stanley Chi for his mentor and help, and also all lab members in the Chi lab. And also would like to thank Professor W. Moller and the Moller lab members for the help on the advanced microscope and how to analyze those imaging trajectories. And also would like to thank the help for for Professor Andrew Speckwith on the help with mathematical modeling, and for Professor Xiaoqiu Lin and her lab for the help on the bioinformatics and Oligo-LiveFISH website. And I also would like to thank 4DN for giving me this opportunity, and thank all the funding agency, and thank you for your attention.